Okay, now, the last part of the day, we are tired enough, but we can understand just the description of organelles that we find in the cytoplasm of eukaryotic cells. In this picture, that you already know because it's the third or fourth time that I show you, you appreciate in the cytoplasm of eukaryotic cell several organelles. When I say organelles, I mean structure enveloped by membranes. And why these are important? The story that I will tell you tomorrow is the traffic of vesicle that we find in cytoplasm, traffic that involve all these organelles: smooth, rough reticulum, Golgi apparatus, and lysosome. We start with the smooth endoplasmic reticulum. Smooth endoplasmic reticulum is constituted by a series of tubules that are present in the cytoplasm. The main function, and in contrast here, pictures at EM level of rough reticulum, and rough reticulum. The difference between smooth and rough reticulum from the morphological point of view is that in rough reticulum we find ribosome stuffed on the surface. Smooth reticulum don't show any ribosome. And depending on the activity of cells, we can get more developed smooth reticulum or rough reticulum. The function performed by smooth endoplasmic reticulum are synthesis, presence of cytochrome P450, synthesis of phospholipids, glycogen degradation in the liver, detoxification in the liver, and Ca+2, calcium ions, storage. The main function is lipid synthesis. Lipid synthesis occurs at membrane level. This is a draw showing a smooth membrane. In this membrane, lipids are synthesized. There are enzymes involved in the synthesis of lipids. And it is interesting to remark that studying lipid synthesis in smooth reticulum, flipases have been found. You remember what flipases are enzymes involved in the flip-flop movement. And this is interesting because lipids undergo to a flip-flop movement in smooth endoplasmic reticulum membrane. Once produced, this lipid can move toward other organelles like as mitochondria or chloroplast in plant cells, or are incorporated in vesicle and can reach Golgi apparatus where they can be modified and reach the external environment of the cell, outside the cell. So the main function is lipid synthesis. Detoxification is a reaction that occurs in the liver cells. This reaction can occur just adding OH group to substances that are hydrophobic. When you add OH group, hydrophobic molecules are converted, becoming hydrophilic, and can be eliminated by urine. In case of intoxication by drugs, this reaction is performed. How this reaction can occur? In the membrane of smooth endoplasmic reticulum, there is present cytochrome P450. This cytochrome recognizes the substrate that should be modified, hydroxylated, and in this reaction that use NADPH, cytochrome P is able to add an OH group to the substrate that this way can be eliminated by urine. The third function played by the smooth endoplasmic reticulum is storage of calcium. Perhaps you know that calcium concentration in the cytoplasm is very low, but in some cases it is important that this concentration increase. When calcium is needed to the cytoplasm, it is released from smooth endoplasmic reticulum. Smooth, rough endoplasmic reticulum. The rough endoplasmic reticulum, since it shows ribosomes stuffed on the surface, it is clear that it is involved in protein synthesis in some way. But this story I will tell you tomorrow because it's difficult to understand at 5 o'clock in the afternoon. But for this reason, I skip this part and we will go back on. Golgi apparatus is another organelle that is present in the cytoplasm. Golgi apparatus is composed by cisterns piled, stacked together. And we have to imagine, the draw is more easy to understand. Each cistern can be considered as a laboratory specialized to perform, to carry out some reaction. In one pile, in one, in Italian it's called dittiosoma. In one stacked cisterns, we can find the cistern that are more close to the rough reticulum. I am tired too. The schist part of the Golgi apparatus, the median part, the median cisterns, and the trans-Golgi network cistern. So we have 3 parts that can be identified in this organelle. The first cistern we find are able to collect the vesicle coming from the rough reticulum. And each cistern is different from one another. The trans-Golgi network sister release vesicle that can reach lysosome, that can reach plasma membrane, that can contain material that should be secreted out of the cell. The trans-Golgi network sister are those projected towards the plasma membrane. From this part of the Golgi apparatus, vesicles are released, and this vesicle can reach the lysosomes, plasma membrane, or can contain materials that should be secreted out of the cell. There are several destinations. As I told you, Golgi complex is not uniform in its composition. We can imagine that, as for instance, in the first cisterns, biosynthesis of sugars that can be added, I cannot say more because you don't know what happens in the rough endoplasmic reticulum. But each part is specialized to perform, to carry different reactions. It is involved in the modification, in post-translational modification of proteins that have been synthesized by ribosomes stuffed on rough reticulum. And is involved also in acrosome formation. Acrosome is a structure that we will find in sperm cell, in mature sperm cell. And is a structure containing lytic enzymes that are useful during fertilization process. Several modifications can occur in the Golgi apparatus. An example is proteolytic cut. It starts in trans-Golgi network. It is completed in a secretion vesicle. As for example, insulin, you know, insulin is a proteic hormone. It is synthesized as a long chain. In the Golgi apparatus, these 30 residues of amino acid are removed to get the mature form of the hormone. That is produced as precursor and is maturated in Golgi apparatus with the elimination of a small segment of 30 amino acid. Another modification involves the post-translational glycosylation. The co-translational glycosylation occurs in the rough endoplasmic reticulum. In Golgi, there are modifications of this chain of sugars, and only some sugars are substituted by others. And the third element of the story is represented by lysosomes. Lysosomes are organelles that we find in the cytoplasm. And in these structures are concentrated lytic enzymes. Lysosomes are the stomach of the cell. Are the structure where is it possible to degrade all the structures, as for example, a mitochondria that don't work at best can be eliminated by lysosome. And the lysosomes are also involved in the degradation of material that comes from the environment. In a lysosome, the pH is maintained at low levels. It's an acid pH, it's about 5. And to keep the pH at this concentration, at this value, there is a pump that allows the H plus to come inside to keep low the level of pH. The content is of a lysosome is acid hydrolase enzymes that are active at acid pH. And to keep low this pH, this protonic pump allows the entrance of H plus into the lysosome. How they work? Something and no, this is lysosome, they can attach the mitochondria that don't work at best and degrade it, or can attach other substances that should be eliminated, degraded and eliminated, are the stomach